This video goes straight into one of the strangest intersections I have come across in my research. It involves the pineal gland, microscopic calcite crystals, piezoelectricity, and the possibility that physical changes in the environment can be transduced into biological effects. This video I think you will really enjoy, so let's get right to it. In a previous video, we looked at a question that sounds strange at first. Could changes in Earth's magnetic field strength affect the human body? Now, one of the studies we discussed was a 2019 eNeuro paper, which reported that specific rotations of an Earth strength magnetic field that it was associated with drops in the alpha band brain activity. And importantly, that response was not random. It depended on the direction and orientation of the field. So, at minimum, that study raised the possibility that the human brain may be more sensitive to magnetic field changes than we usually assume. Now, later in that same video, we looked at a different but related question: whether changes in geomagnetic activity can be associated with physiological changes in the body. One of the findings we discussed was that higher geomagnetic activity was linked to lower levels of a melatonin metabolite. This was measured through overnight urinary 6-hydroxymelatonin sulfate excretion, and that effect appeared with a delay of roughly 15 to 33 hours. Melatonin is one of the body's core hormones for circadian timing and sleep regulation. A deeper question really emerged for me. I essentially wanted to know, how is it that something from outside the brain, changes in the magnetic field strength or orientation, could impact hormone production inside? Luckily, I was not the only one asking this question. There are already a few proposed pathways in the literature. One possibility is induced electric field effects. Even weak magnetic field changes can, in principle, produce very small electrical effects in tissue, and those may influence things like neuronal excitability or calcium signaling. Another possibility involves circadian protein sensitivity, especially proteins like cryptochromes. These are already tied to biological timing and in some contexts have been discussed as possible magnetically sensitive molecules. A third possibility is the autonomic pathway. Melatonin synthesis is regulated through sympathetic signaling to the pineal gland. So, if magnetic conditions influence circadian or autonomic regulation, that could potentially alter melatonin output downstream. Now, to be clear, I'm not presenting any of these as confirmed explanations. These are only proposed mechanisms, not settled ones. But while I was looking into this question, I came across a fourth possibility that surprised me enough that I had to follow it further. But before getting into the two papers themselves, let's briefly ground this in the star of the show, the pineal gland. The pineal gland is a small neuroendocrine gland in the epithalamus, near the center of the brain, and it is only about 5 to 8 millimeters across. It produces melatonin, especially in darkness, and it plays a central role in circadian timing and sleep-wake regulation. It also influences broader neuroendocrine and seasonal rhythms. Now, the topic of pineal crystals is often discussed in a much broader way. I am not making a broad claim about what these crystals mean. My focus here is actually pretty narrow. If crystalline material is present in the pineal gland, could it do more than simply sit there? Could it convert or transduce a change in the external magnetic environment into some kind of physical effect inside the gland? This brings us to the 1996 paper, Piezoelectricity in the Human Pineal Gland. What interested me immediately is that the authors were asking the same question: if electromagnetic conditions can influence melatonin-related biology, how exactly is it doing this? They already knew that the pineal gland contained calcified crystalline structures, but not all of them looked the same. Some are larger and mulberry-like, commonly called brain sand. Others were smaller, faceted, and geometric. Many of the smaller crystals were only a few micrometers across. They wondered whether some of these structures might do more than simply exist inside the gland. Could they act as physical transducers? Could an external field be transduced into a physical effect within the gland? So the first step in this paper was to isolate these crystal types, and begin to determine if this idea held any weight. One consideration here is scale. Many of the crystals that they found were extremely small. Now, as you can imagine, that created a practical problem. If you want to know whether a material is piezoelectric, the most direct test would be to apply stress to it and see whether it produces a voltage. But these crystals were too small for that kind of direct test, so the researchers used an indirect method instead. They turned to second harmonic generation, or SHG. Here is the basic idea. They shine laser light of a known frequency into a crystal, and then they ask whether the crystal produces a new light signal at exactly twice that frequency. Because frequency and wavelength move in opposite directions, that also means the new light would have half the wavelength. If that double frequency signal appears, it is what we call positive SHG. SHG is basically a way of asking whether the crystal has the kind of internal asymmetry that often goes along with piezoelectric behavior. More specifically, SHG is associated with crystals that are non-centrosymmetric, meaning their structure does not look the same in opposite directions around a center point. Crystal symmetry is tightly linked to piezoelectricity. There are 32 crystallographic point groups in total. 21 are non-centrosymmetric, and 20 of those 21 are piezoelectric. So a positive SHG result does not directly prove that a crystal is piezoelectric, but it does make it a very strong candidate, giving it a 95% chance. So at this point, the earlier work was suggestive, but not complete. The researchers were finding a consistent SHG signal in all the pineal samples they examined, and that signal disappeared with heating, which supported the idea that something crystalline in the gland was responsible for producing it. Since SHG points towards a non-centrosymmetric structure, and since most non-centrosymmetric crystals are piezoelectric, this made the pineal microcrystals very interesting candidates. But there was still a major unresolved problem. They had evidence of a signal, but they still did not fully know what the microcrystals actually were. And to me, that is the main question they were trying to answer in the follow-up paper. The first paper was essentially saying there appears to be a small crystalline population in the pineal gland with structural properties that make piezoelectricity plausible. But until you know the identity of those crystals, you do not really know how seriously to take that possibility or how to think about their behavior. The larger, more familiar mulberry-like concretions were already understood to be hydroxyapatite rich. Hydroxyapatite is a calcium phosphate mineral and is also the main mineral found in bones and teeth. So that part was not really the mystery. The mystery was the smaller crystal population. Those were the crystals generating the real interest because they appeared structurally different, they were much smaller, and they were better candidates for explaining the SHG signal. So the next step was obvious. They needed to characterize the microcrystals themselves. In the later work, they reported that these microcrystals were present in every pineal gland they examined, across subjects ranging from age 15 to 68. They estimated roughly 100 to 300 crystals per cubic millimeter of gland tissue. They also emphasized that these were much smaller than the classic larger pineal concretions and that they appeared in a few recurring shapes, including cubic, hexagonal, and cylindrical, with the cylindrical form dominating the samples. Now, the picture was becoming more specific. The pineal gland did not just seem to contain one kind of calcified structure. It appeared to contain at least two distinct populations, the larger hydroxyapatite-rich concretions that had already been recognized, and a much smaller population of microcrystals that still needed to be identified. So the second paper was not just a repetition of the first. It was a necessary next step. If the first study raised the possibility that these structures might have unusual physical properties, the follow-up study had to answer a simpler but essential question first. What exactly are these crystals? In order to identify what those crystals actually were, they approached it from several angles. First, the elemental composition matched calcium carbonate, meaning the crystals contain calcium, carbon, and oxygen, in the proportions you would expect. Second, the crystal structure matched a calcite lattice when they looked at it with the electron diffraction. And third, the Raman spectral signature also matched calcite. And this is what you're seeing here in the spectrum comparison, where the pineal microcrystals line up with the calcite reference. So by this point, the case had become much stronger that these pineal microcrystals were calcite. The SHG reassessment is what makes this especially interesting. The larger hydroxyapatite rich concretions, the more familiar brain sand structures, did not show SHG. The calcite associated microcrystals showed a weak SHG signal, and the pineal tissue signal matched the calcite pattern rather than the hydroxyapatite one. Now that solved one problem, but immediately created another. On the one hand, the researchers now had a much clearer identification of the smaller crystal population. But on the other hand, ideal pure calcite is not generally treated as piezoelectric. This created a bit of tension. Earlier, the SHG findings suggested a non-centrosymmetric structure, which made piezoelectricity plausible. But now the crystals appear to be calcite, and ideal calcite should not behave that way. It should not be piezoelectric. So, how do these two things fit together? Why would something identified as calcite still show a weak SHG signal? Those earlier images revealed the microcrystal's rough exterior, and this provided just the clue they needed. The proposed answer here is something called twinning. Twinning means that different parts of a crystal can grow in different orientations within the same overall structure. Instead of one perfectly uniform crystal, you end up with internal domains and boundaries. Importantly, those internal boundaries can locally break symmetry. So even if the crystal, in a broad, idealized sense, is calcite, the actual biological version may not be growing as a perfectly uniform crystal. Therefore, local symmetry breaking could allow for piezoelectric behavior to emerge. Sounds reasonable, right? They were able to point to an additional example. The microcrystals they reported also resemble Otoconia of the inner ear. What is interesting is that Otoconia are also calcite, and they have already been reported to exhibit piezoelectric behavior. So this does not prove with 100% certainty that pineal microcrystals are doing the exact same thing, but it does make the broader idea feel way more possible. It suggests that there may already be biological examples where calcite-based structures grown in real tissue rather than in perfect lab conditions behave in more complicated ways than the ideal crystal model would suggest. So, what do I take from these two papers? To me, the first striking point is that the pineal gland may contain a small population of microcrystals with unusual physical properties that are still not part of the way most people think about the organ. The first paper raised some possibility that some of these crystals may have structural features consistent with piezoelectric behavior. The second paper strengthened the case that these microcrystals are calcite, while also opening the door to a more biologically realistic picture in which calcite and living tissue may not behave like a perfect crystal grown in a lab. If their reasoning here is right, and it looks pretty well done, these pineal microcrystals may act as transducers. Then suddenly, changes in external magnetic environment could, in principle, be converted into physical effects inside the gland itself, with possible downstream consequences for local cellular signaling and hormone production. At the very least, I think these papers point to a real and underexplored biophysical question, and I am certainly curious what you think about it. Thank you so much for your time, and until I see you on the next one, stay curious and take care.